Welcome! Today we will explore the fascinating world of molecular biology by examining the structure of DNA, RNA, and proteins, the fundamental molecules of life. DNA serves as the blueprint of genetic information, RNA acts as the messenger and translator, and proteins carry out essential cellular functions. Understanding their structures will help us uncover how genetic information is stored, expressed, and utilized in all living organisms. These are our learning targets for the lesson. First, I can describe building blocks of DNA, RNA, and proteins. Next, I can discuss the different levels of protein structure, primary, secondary, tertiary, and quaternary. Next, I can identify the structural and functional differences between DNA and RNA, and I can explain how protein structural features may influence their functions. Biomolecules are the essential chemical compounds that sustain life, playing a crucial role in the structure and function of all living organisms. They include carbohydrates, lipids, proteins, and nucleic acids, each serving a unique purpose in biological systems. These molecules are responsible for energy storage, genetic information transfer, cellular structure, and metabolic processes. Biomolecules are primarily composed of carbon, hydrogen, oxygen, nitrogen, and phosphorus, forming complex structures that interact in intricate biochemical pathways. Understanding this is fundamental to fields such as biochemistry, molecular biology, and medicine, as they underpin all physiological processes in living organisms. Now, think of monomers as small building blocks like Lego pieces. When these small blocks connect, they form a larger structure. This bigger structure is called a polymer. For example, monomer is a single bead in a necklace. Our polymer would be a full necklace made by linking many beads together. In biology, monomers and polymers make up important biomolecules. For sugars, the monomer sugar, carbohydrates are the polymers like starch. In amino acids, which are monomers, it creates the proteins. Proteins are the polymers like enzymes. Nucleotides are the monomers. It creates DNA and RNA, or the polymers of nucleic acids. They carry genetic information. Nucleic acids are large complex molecules that store and transmit genetic information in living organisms. They are essential for life because they direct the production of proteins and regulate cellular activities. The two main types of nucleic acids are DNA or deoxyribonucleic acid and RNA or ribonucleic acid. What are the differences between DNA and RNA? Let's take a look at it. For its form or feature, DNA is double-stranded or double helix, while RNA is single-stranded. In its sugar, for DNA, it's deoxyribose, and for RNA, it's ribose. Therefore, their names deoxyribose or deoxyribonucleic acid and ribonucleic acid respectively. The primary difference between ribose and deoxyribose lies in the presence of a hydroxyl group at the second carbon in ribose, while deoxyribose has a hydrogen atom at the same position. For bases or nucleotide bases, for DNA, there's adenine, thymine, cytosine, and guanine. For RNA, there's adenine, uracil that replaces thymine, cytosine, and guanine. For its function, DNA stores genetic information, stores RNA and protein encoding information. It transfers the information also to the next generation of cells, while in RNA, it helps in protein synthesis and gene regulation. For its stability, DNA is more stable and resistant to degradation, while RNA is less stable and breaks down more easily. Nucleic acids are composed of smaller units called nucleotides. Nucleotides are the monomers of nucleic acids, which consist of three components: a phosphate group, a sugar molecule, it's deoxyribose in DNA and ribose in RNA, and a nitrogenous base, adenine, thymine, cytosine, guanine in DNA, and uracil replaces thymine in RNA. Now, let us take a closer look into it. Purines and pyrimidines are two types of nitrogenous bases that makes up the building blocks of DNA and RNA. The key difference between them lies in their structure. Purines have a double ring structure, making them larger, while pyrimidines have a single ring structure, making them smaller. The purine bases include adenine and guanine, whereas the pyrimidine bases include cytosine and thymine, found only in DNA, and uracil, found only in RNA. In DNA and RNA, purines always pair with pyrimidines to maintain a stable structure. Adenine pairs with thymine or uracil in RNA, and guanine pairs with cytosine through hydrogen bonding. This complementary base pairing is essential for genetic information storage, replication, and protein synthesis. The key difference between the sugars in DNA and RNA lies in their structure and function. DNA contains deoxyribose while RNA contains ribose. Both are 5-carbon sugars, but deoxyribose lacks an oxygen atom at the second carbon, whereas ribose has a hydroxyl group at the same position. This small structural difference has significant effects on the stability and function of these molecules. Because deoxyribose lacks the extra oxygen, DNA is more stable, making it well suited for long-term genetic storage. In contrast, the presence of the hydroxyl group in ribose makes RNA more reactive. As we all know, oxygen is highly reactive and less stable, which is ideal for short-term genetic functions such as carrying message for protein synthesis using mRNA. This difference in sugar structure is one of the reasons DNA serves as the permanent genetic blueprint, while RNA acts as a temporary messenger in the cell. The sugar phosphate backbone is the structural foundation of DNA and RNA, providing stability and support to these molecules. It is composed of alternating sugar molecules, deoxyribose in DNA and ribose in RNA, and phosphate groups, which are linked together by what we call phosphodiester bonds. This backbone forms a continuous chain that holds the nucleotide sequence in place. The phosphate groups give the backbone a negative charge, making DNA and RNA hydrophilic or water-attracting and allowing interactions with proteins. The backbone also establishes the 5' to 3' directionality, meaning nucleotides are connected in a specific order, which is crucial for processes like DNA replication and RNA transcription that we're going to know later on. While the sugar phosphate backbone provides strength and stability, it does not carry genetic information. Instead, the nitrogenous bases attached to the sugars extend outward and participate in base pairing in DNA. This strong yet flexible structure ensures the integrity of genetic material and its ability to function properly in cells. As mentioned in the previous slide, two nucleotides are bonded together by a phosphodiester bond. Where is the bond located? It links the phosphate group of one nucleotide and the sugar of another molecule. If you're going to untwist the DNA, it will form a ladder. The base of the ladder is the phosphate sugar backbone, and the connections between the nitrogenous bases of two strands of DNA connected by hydrogen bonds serves as the rungs of the ladder. The major and minor grooves are structural features of the DNA double helix that result from the way the two strands twist around each other. These grooves form because the sugar phosphate backbone are not evenly spaced, creating alternating wider and narrower gaps along the DNA molecule. The major groove, as you can see in the picture, is the larger of the two and occurs where the backbones are farther apart. It is more accessible to proteins, making it the primary site for protein binding, such as transcription factors that regulate gene expression. The major groove also contains more chemical information, allowing proteins to recognize specific DNA sequences. In contrast, the minor groove is the smaller groove, forming where the backbones are closer together. While it is less accessible for protein binding, some small DNA binding proteins and certain drugs interact with it. These grooves play a crucial role in DNA function by allowing proteins to interact with the genetic material or the DNA without unwinding the helix. This structural feature is essential for gene regulation, DNA replication, and repair in living organisms. Now, base pairing in DNA follow the Chargaff's rule, where specific nitrogenous bases always pair with each other through hydrogen bonds. In the DNA double helix, adenine pairs with thymine using 2 hydrogen bonds as shown in the diagram, while guanine pairs with cytosine using 3 hydrogen bonds. This complementary base pairing ensures the stability of DNA structure and allows for accurate replication and transmission of genetic information. The base pairing follows a purine-pyrimidine rule, meaning a larger purine, it's either A or G or adenine or guanine, always pairs with the smaller pyrimidine, the thymine or cytosine or C or T. This keeps the DNA structure uniform and maintains the double helix shape. The hydrogen bonds between bases provide enough stability to hold the strands together while still allowing them to separate during DNA replication and transcription. The two strands of DNA are linked together by hydrogen bonds between paired bases. Pairing a purine with a pyrimidine results in a uniform diameter, as you can see in the picture, for the helical shape of DNA. If a purine A or G were to pair with another purine, take a look at the picture, it would cause structural instability in the DNA double helix. Normally, DNA follows the purine-pyrimidine rule, where a larger purine pairs with a smaller pyrimidine to maintain a uniform helix width. However, if two purines paired together, the DNA structure would be disrupted. Since purines are larger, the double-ringed molecules, their pairing would create a bulge in the DNA strand, distorting the helical shape. Additionally, normal base pairing is stabilized by specific hydrogen bonds. Adenine to thymine forms 2 bonds, guanine to cytosine forms 3. A purine-purine pairing would not form stable hydrogen bonds, weakening the overall DNA structure. This instability can lead to errors during DNA replication, increasing the likelihood of some mutations that may cause genetic disorders or disrupt essential biological functions. To prevent these issues, DNA polymerase, the enzyme responsible for DNA replication, has proofreading mechanisms that detect and correct improper purine-purine pairings. These will ensure that DNA maintains its stable, double helix structure, allowing for accurate replication and proper cellular function. DNA-based pairing is fundamental for the accurate storage and transmission of genetic information. The specific pairing of nitrogenous bases ensures that genetic instructions are precisely copied during DNA replication and accurately transcribed into RNA. This process is crucial because DNA serves as the blueprint for building proteins, which carry out essential functions in the body. Let us talk about the functions of proteins. Enzymatic proteins are a special type of protein that acts as biological catalysts, meaning they speed up chemical reactions in the body without being consumed in the process. These proteins are essential for life because they help regulate and accelerate vital biochemical reactions that would otherwise occur too slowly to sustain life. Each enzymatic protein has a specific active site, a uniquely shaped region where a substrate binds. This interaction follows the lock and key model or induced fit model, ensuring that enzymes work only with specific substrates. Defensive proteins are a type of protein that help protect the body against harmful invaders such as bacteria, viruses, fungi, and toxins. Their primary role is to support the immune system by identifying, neutralizing, and eliminating threats to maintain overall health. One of the most important defensive proteins is antibodies, which are produced by B cells in response to infections. Antibodies recognize specific antigens or foreign molecules on pathogens and either neutralize them directly or mark them for destruction by other immune cells. For example, when a virus enters the body, antibodies attach to it, preventing it from infecting healthy cells. Storage proteins are a type of protein that serve as reserves of essential nutrients such as amino acids, minerals, and metal ions, which can be used by the body when needed. These proteins are crucial for growth, development, and metabolism in both plants and animals. Last, transport proteins are specialized proteins that help move molecules across cell membranes or throughout our body. Since many essential substances such as oxygen, nutrients, and ions cannot pass through cell membranes on their own, transport proteins ensure the proper movement, maintaining cellular and overall body function. And there's a lot more functions. Let's go to the next one. Hormonal proteins are signaling molecules that regulate various biological processes by acting as chemical messengers between cells, tissues, and organs. These proteins help maintain homeostasis or the balance in the body by controlling functions such as growth, metabolism, reproduction, and stress response. One key example is insulin, a hormonal protein that regulates blood sugar levels by signaling cells to absorb glucose for energy. Next, receptor proteins. These are specialized proteins found in cell membranes or inside cells that help receive and transmit signals from the environment or other cells. They play a crucial role in cell communication, allowing cells to respond to hormones, neurotransmitters, and other signaling molecules. These proteins have specific binding sites that recognize and attach to signaling molecules, triggering a response inside the cell. Next, contractile and motor proteins are specialized proteins that generate movement in cells and muscles. They are essential for functions such as muscle contraction, cell division, and intracellular transport. Among these proteins are actin and myosin as shown in the picture. These are the most well-known and plays a key role in muscle contraction and cellular motion. Actin is a thin, filamentous protein that forms long chains and serves as structural framework inside the cells. It is involved in cell shape, movement, and division. In muscle cells, actin filaments create a scaffold for myosin to interact with during contraction. Now, myosin is a thicker motor protein with a unique structure that allows it to walk along actin filaments. Myosin has head regions that bind to actin and using energy from ATP or adenosine triphosphate pull the actin filaments closer together. This repeated interaction between actin and myosin is what causes the muscle contraction and enables movement such as walking, blinking, and even our heartbeats. Next, structural proteins are essential for providing support, strength, and shape to cells, tissues, and organs. Two of the most important structural proteins are keratin and collagen, as shown in the picture, which help maintain the integrity of the body's structures. Keratin is a tall, fibrous protein found in skin, hair, nails, and horns of animals. It provides protection and durability, making these structures resistant to damage, water loss, and environmental stress. In skin, keratin forms the outermost layer or epidermis, acting as a protective barrier against bacteria and toxins. Collagen is the most abundant protein in the body and serves as the main component of connective tissues such as tendons, ligaments, cartilage, and skin. It gives tissues strength, elasticity, and structure, helping them withstand stretching and pressure. In skin, collagen maintains firmness and flexibility, while in bones and joints, it provides structural support and shock absorption. The monomer of proteins is an amino acid. Amino acids are small organic molecules that serve as the building blocks of proteins. There are 20 different amino acids, each with a unique structure, but they all share the same basic components. First, an amino group. a carboxyl group, a hydrogen atom, and a variable R group or side chain attached to a central carbon atom. The R group determines the properties and function of each amino acid. Amino acids link together through peptide bonds in a process called dehydration synthesis, forming long chains known as polypeptides. Polypeptides then fold into specific 3D shapes to become functional proteins. The sequence and arrangement of amino acids will determine a protein structure and function, influencing everything from enzyme activity to muscle contraction. These are the 20 amino acids. If you're going to take a look at it, the variable R group or the side chain attached to the central atom is the one that determines the properties of each of the amino acids. Amino acids serve as the building blocks of proteins, but their true function comes from how they are arranged and folded into complex structures. When amino acids link together through peptide bonds, they form polypeptide chains. However, a protein's function is not determined solely by its sequence of amino acids. It depends on its 3D structure. This hierarchical organization allows proteins to perform diverse functions, from catalyzing reactions or like enzymes to providing structural support like collagen and keratin. Without those proper folding, proteins may lose their function, leading to diseases. Thus, the transition from amino acids to protein structures is essential for life, ensuring proteins carry out specific biological roles. The primary structure of a protein refers to the linear sequence of amino acids in a polypeptide chain. This sequence is determined by the genetic code DNA and dictates how the protein will fold and function. Each amino acid in the chain is linked to the next by a peptide bond, forming a long chain called polypeptide. The specific order of amino acids in the primary structure is crucial because it directly influences the higher levels of protein structure, the secondary, tertiary, and quaternary. Even a small change in the sequence, such as a single amino acid substitution, can affect the protein's function. The secondary structure of a protein refers to the local folding patterns formed by the polypeptide chain due to hydrogen bonding between the backbone atoms. These structures provide stability and serve as the foundation for protein's overall shape. The two most common secondary structures are the alpha helix and the beta-pleated sheet. In alpha helix, the polypeptide chain coils into a spiral shape, stabilized by hydrogen bonds between the carbonyl group of one amino acid and the amide group of another amino acid located 4 residues away. This structure is commonly found in fibrous proteins like keratin, which strengthens hair and nails. In beta-pleated sheet, the polypeptide chain folds back and forth, forming parallel or anti-parallel strands connected by hydrogen bonds. This arrangement creates a strong, flexible structure commonly found in proteins like silk fibrin. The secondary structure plays a crucial role in determining how a protein will further fold into its tertiary structure. Now, disruptions in this level of structure can lead to improper protein folding. Next, the tertiary structure of a protein refers to its 3D shape, which results from the further folding of the secondary structure. This unique shape is stabilized by various interactions between the R groups or the side chains of amino acids, including hydrogen bonds, disulfide bonds, ionic interactions, and hydrophobic interactions. The tertiary structure determines the function of a protein, as its specific shape allows it to interact with other molecules effectively. Different types of bonds and interactions contribute to the stability of a tertiary structure protein. First one, hydrogen bonds form between polar R groups, helping to maintain the structure. Next, disulfide bonds or covalent bonds between sulfur atoms and cysteine residues provide strong structural support. Next, ionic bonds occur between positively and negatively charged R groups, and hydrophobic interactions cause non-polar amino acids to cluster towards the protein's interior, avoiding water. The tertiary structure is essential for a protein's biological activity. For example, enzymes rely on their specific shape to bind to substrates, and antibodies must maintain their structure to recognize antigens. If the tertiary structure is disrupted due to mutations or environmental factors like high temperature or pH changes, the protein may become denatured, losing its function. Last, the quaternary structure of a protein refers to the arrangement and interaction of two or more polypeptide chains or subunits to form a functional protein. While not all proteins have a quaternary structure, those that do rely on multiple subunits working together to perform their biological functions. These subunits can be identical or different and are held together by hydrogen bonds, ionic interactions, hydrophobic forces, and sometimes disulfide bonds. The quaternary structure is crucial because it enhances protein stability, functionality, and regulation. If the subunits do not assemble correctly, the protein may not function properly, leading to diseases. For instance, in sickle cell anemia, a mutation in the hemoglobin subunits causing them to stick together abnormally, affecting the oxygen transport. Now, understanding the quaternary structure is essential in biotechnology, medicine, and drug development, as many therapeutic proteins such as antibodies and insulin rely on precise subunit interactions to be effective. Sickle cell disease or SCD is caused by a mutation in the gene that encodes hemoglobin, the protein responsible for carrying oxygen in red blood cells. Normally, hemoglobin is made up of 4 polypeptide subunits, 2 alpha and 2 beta chains, and has a flexible, round shape, as you can see in the picture, that allows red blood cells to move smoothly through blood vessels. In sickle cell disease, a single mutation in the beta-globin gene or HBB causes the amino acid glutamic acid to be replaced with valine at position 6 of the beta chain. This altered hemoglobin is called hemoglobin S. Unlike normal hemoglobin, the hemoglobin S tends to stick together under low oxygen conditions, forming long, rigid fibers inside red blood cells. This causes the cells to change from their normal round shape into a crescent or sickle shape. These sickle-shaped cells have several harmful effects. First one is reduced oxygen transport. Sickled cells do not carry oxygen as efficiently as normal red blood cells. Next one is blockage of blood flow. Due to the rigid shape, sickled cells can clump together and block small blood vessels, leading to painful crisis, organ damage, and increased risk of stroke. Shortened cell lifespan also is another harmful effect because normal red blood cells live about 120 days, but sickled cell breaks down in just 10 to 20 days, leading to anemia or a shortage of red blood cells. The structure and function of hemoglobin highlight the importance of proper protein formation, as even a single amino acid change can lead to disorders like sickle cell disease. This emphasizes the critical role of protein synthesis, the process by which cells create proteins based on genetic instructions. Protein synthesis is the one that you can see in the diagram. Proteins like hemoglobin are built through a highly regulated, multi-step process involving DNA, RNA, and ribosomes. This process ensures that proteins are made accurately, allowing them to perform essential functions such as oxygen transport, enzyme activity, and structural support. Understanding protein synthesis provides insight into how cells translate genetic information into functional molecules and how errors in this process can lead to diseases. Protein synthesis is a biological process by which cells produce proteins based on genetic instructions stored in DNA. It occurs in two main stages, transcription and translation. Transcription is from DNA to RNA. In the nucleus, as you can see in the picture, an enzyme called RNA polymerase reads a gene's DNA sequence and creates a complementary messenger RNA or mRNA strand. This mRNA carries the genetic code from DNA to the ribosome for protein assembly. Now, for translation process, that's RNA to protein. In the cytoplasm, the mRNA attaches to a ribosome where it is read in sets of 3 bases called codons. Each codon corresponds to a specific amino acid which is delivered by a transfer RNA or tRNA. The ribosomes will link the amino acids together, as you can see in the picture, in the correct sequence to form a polypeptide chain which then folds into functional protein. This process ensures that cells produce the proteins necessary for growth, repair, and various biological functions. Error in protein synthesis can lead to diseases, making this process essential for life. This is a summary of the things we talked about today. On the first box is the structure of a nucleotide, monomers of nucleic acids. Next is the main difference between two types of nucleic acids, the DNA and the RNA. DNA is a double helix structure that stores hereditary information, contains deoxyribose, and the nitrogenous bases adenine, thymine, cytosine, and guanine. RNA, however, is a single-stranded structure that performs various functions in gene expression and contains adenine, cytosine, guanine, and uracil as nitrogenous bases. We also talked about the monomers of amino acids, the functions of proteins, and the different shapes and structures of proteins. Now, let us recall our learning targets. Okay, I can describe the building blocks of DNA, RNA, and proteins, discuss the different levels of protein structure, identify the structural and functional differences between RNA and DNA, and explain how protein structural features may influence their functions. We were able to learn each of the learning target stated. That is all for this lesson. Thank you for listening and see you on the next one.